Hello Year 12, welcome to another flipped video for biology, for maintaining a balance. Today we're looking at 2 dot points that cover how organisms live in salty environments. The main concept that we'll be looking at is the first dot point is called Enantiostasis, and there are a number of processes in our second dot point that we'll be looking at that has to do with how plants survive in salty regions. So let's get stuck into this. The first one is, the first dot point has a define verb in it. That basically means, almost word for word, you need to understand the definition of enantiostasis as the maintenance of metabolic and physiological functions in response to variations in the environment. We then need to be able to discuss this in terms of its importance to estuarine organisms. So estuarine organisms are organisms that live in estuaries, and we'll get to cover what they are in a moment. And the context for this is that they use enantiostasis in order to maintain appropriate salt concentrations. So let's put enantiostasis in context. Some organisms live in the environments where they experience extreme fluctuations in conditions. These plant and animals have evolved adaptations that allow them to cope physiologically with these fluctuations. This is called a survival mechanism, enantiostasis. We've got the definition of it just down here, so if you haven't got that one down yet, I suggest you get that one into your notes. But the survival of a species in an estuary, where salt and water concentrations fluctuate broadly on a daily basis, depends on their ability to either avoid or tolerate them. So the two concepts that we're looking at is this avoidance and toleration. It's important to note here that enantiostasis is not just about salt levels. It's any factor of the environment that changes frequently. One example that we might be familiar with is that pressure builds as you go deeper into the ocean, and it's about 1 atmosphere's worth every time that you go down 10 meters. So birds who are diving into the water need to be able to cope with those sudden changes of pressure, and if they dive particularly deep, you've got a significant pressure gradient that they have to be able to cope with. Humans don't have an adaptation to handle this pressure gradient, and it results in a condition that we call the bends. Scuba divers are very familiar with it, and it's a major factor in getting your scuba diver license. You must know how to kind of get to the top of the water body without going so fast that you cause issues for your body. Alright, so our focus today is going to be looking at the impact of tides on water. In high tide, seawater flows into the river mouth, creating an environment with a high salt concentration than the cytoplasm of cells and body fluids in the organisms that live there. This salt water has the tendency to draw water out of cells via osmosis. During low tide, fresh water is entering the estuary from the rivers as the water is receding in the ocean, salt water is receding in the ocean, and we get an environment where the outside of the cell has a lot more fresh water than the inside, and that causes water to move into the cells via osmosis. We can describe this as, if we think about water in terms of its density, salt water is more dense than fresh water, and so that fresh water is always going to sit on top of the salt water where it can. So in high tide, if we use our little crab here as an example, it is sitting in a solution that has 35% salt in it, which is a very salty water. And then over here in the low tide, this crab is sitting in a solution of 15% salt water. You can see that in high tide that wedge is quite steep, and in low tide it's quite shallow. This represents kind of the gradient change, so this is a very quick change from this point here all the way up to here. That is a very quick change in terms of the amount of distance, the distance in the water body. Whereas if we go from here to here, it's significantly longer in low tide. So let's talk about the animals that are occupying these spaces. What are they trying to do? Well, osmoregulation is the focus of this process. They're trying to make sure that they have the right amount of water for their cells. So, estuaries are challenging environments to live in, and despite the enormous fluctuations, we see organisms living there. So we're looking at one or two ways in which these organisms can survive. It comes back to that avoidance or tolerance of the conditions. The first one, which is focusing on the tolerance, we have a name for it. They are OSMOCONFORMERS. They are organisms that tolerate the changes of the environment by altering the concentration of their internal solutes to match the external environment. Their body fluids conform to that of the environment. This means that their metabolism, the processes that are going on inside of their cells, the chemical processes going on inside their cells, can continue despite the salt increase. If we hark back to the first part of our course, we learnt that those metabolic processes are regulated by enzymes. So really we're talking about what impact does salt have on enzymes, and in this case, salt can inhibit the function of enzymes. So these osmoconformers, they let their internal conditions replicate that of the environment, but they've got some sort of thing that they do that allows them to counteract the impact on these, the impact of extra salt in the cells. And what that is they change their pH. The changing of the pH actually can counteract the increased sodium levels in a cell. So that's a pretty cool adaptation, and you can see here that starfish is an example of an osmoconformer. So is a jellyfish. You know, they're pretty cool, these osmoconformers. So now we've got a big picture of a crab, so you can kind of guess where we're going with this one. Osmoregulators are organisms that have systems in place or adaptations that allow them to avoid changes in their internal environment. They regulate solute concentrations within their body regardless of the differing external environment. How they do this is you can have multiple adaptations. Some of these organisms have shells. Some of them have the ability to close valves to trap seawater. They can burrow in rock crevices and sand, and they can live in rock pores. We'll talk about this ability to close valves to trap seawater. If you're an organism that prefers high salt concentrations in the water, if you can trap some of that salty seawater, when the tide goes out and you're inundated with fresh water, if you're losing some solutes to that fresh water, you can actually replace them with that little supply of seawater that you are taking with you. The shells act as a physical barrier to either salt intake or salt loss, and the burrowing in rock crevices and sand and living in rock pores reduce the surface area of that organism to those harmful conditions or the harmful fluctuations that they encounter. So that's enantiostasis. It's the challenge of it's the process that organisms who are challenged by a fluctuating environment put in place, and it falls into two categories: osmoconformers and osmoregulators. We're now going to look at our second dot point, and it's going to be talking about using evidence to discuss processes used by different plants for salt regulation in saline environments. Trees that live in saline environments are under water stress. Too much salt, as we've discussed, can inhibit enzymes, and therefore salt accumulation is toxic to plants. Any plant that lives in a saline environment is called a halophile. as you can see, we've got a picture here of an estuary. We have the ocean in the background, and then we have the river mouth forming in the centre of the picture there. We're largely going to see these as a majority of our examples, but we also have inland areas where rising water tables can bring natural salts into the soil and up to the surface. We actually have this at our school. We have a dryland salinity problem, so we have some plants on the wetlands that are adapted to a high salt environment. The two examples that you're going to want to remember for plants are mangroves and salt marsh plants or salt bush. These are going to be two handy examples for you to have. So the dot point was asking us to look at some processes, and there are 4. We're going to see the next 2 in a moment. The first process is exclusion. The exclusion is used by a bunch of different types of mangroves, and it's largely because they're generally in a water body where there's lots and lots of salt. And so what this does is that it's a physical barrier that prevents salt from even just entering the roots in the first place, which is a pretty cool adaptation to have. The next one is excretion, and we have, I think in my head it makes this as the most effective adaptation to have. The plant, whether it be a mangrove or a salt bush, has special glands, usually in their leaves, that push salt out, and it crystallizes, and then if it rains or there's spray, it will be dissolved and washes off the plant, so it actually leaves the plant physically, which is a pretty cool adaptation to have. It requires active energy and can be a little intensive in terms of the energy demands on the plant, but the payoff is worth it for the plant's survival in the long term. So the next process is accumulation. It's very similar to excretion, except rather than expelling the salt from the plant pushes the salt into a particular part of it. Usually it's going to be leaves or sometimes bark. And these, once these get high salt levels, they will kind of dry out, and when they dry out they get brittle, and there's a high chance that they will actually just fall off the plant. And if they fall off the plant, they're no longer part of its physiology, and so the plant can continue on. It's a really effective way of getting rid of toxins and salt. Finally, this is a less common example. We have the reduction of transpiration. And if you can remember about the transpirational stream, water will leave the plant and it can enter through the roots. So if you're stopping water from leaving the plant through the leaves, you don't need to pull in water from the roots. And if you're pulling in water from the roots, you'll naturally pull in salts with it. So preventing that is a good strategy to have. The way the plant will do this is often, it's called the drooping of the leaves. If you droop the leaves vertically, you expose less of the plant to light, less photosynthesis occurs, less water is produced, and the stomates stay closed, essentially. Okay, that's it for the presentation. Two pretty straightforward concepts, but one thing that I think Mrs. Skelly and myself will try and do a good job of is in class making sure that we know the difference between enantiostasis and homeostasis, because while they both help the organism maintain metabolic functions, they do them in different ways. Okay, see you guys.